Howdy, my name is Natalie Chavez.
Eros Aranda,
And I'm Micaela Cavazos.
and this is our project on the summary of innate immunity. Oh, how does the immune system respond to microbes and damaged cells? The innate immune system has the ability to recognize microbes and damaged cells through the use of specialized receptors called PRRs or pattern recognition receptors. These receptors will search these cells for common features associated with a large range of cells or pathogen types. Examples of PRRs are TLR or toll-like receptors found on the cell surface or inside the cell. What differentiates this from adaptive immunity is that rather than searching invaders for specific antigens, it will use these standard features to identify the molecules hazardous to the body and will begin the process of destroying it. The two main parts of PRRs are known as PAMP and DAMP. PAMPs, or pathogen-associated molecular patterns, are molecules inside the body that trigger an immune response due to recognizing microorganisms. PAMPs will be recognized by PRRs due to specific features or properties that these pathogens present. Common PAMPs that are found and react to are nucleic acids, membrane proteins, lipoproteins, and flagellin, which are found in gram-positive bacteria. Once these PAMPs have been identified, the innate immune system will mount a response and eradicate the microbe. So in simpler terms, PAMPs are specific features that the body will recognize that signifies that the molecule isn't natural to the body, that it's an invader or it's foreign anti. So the other half of this is DAMPs. DAMPs are damage-associated molecular patterns, and they're very similar to PAMPs in that they're recognized by PRRs in order to mount an immune response. DAMPs are released from cells that are either stressed or injured and may pose a threat or otherwise need to be removed. A very telltale sign that a cell is damaged is the presence of ATP outside the cell. As ATP is vital for intracellular functions, the presence of any ATP outside the cell indicates some present issue. The significant difference between PAMPs and DAMPs is that PAMPs are derived from microorganisms, while DAMPs come from whole cells.
I'll be presenting the different components of innate immunity and their functions. Neutrophils are the first responders that attack to bacteria and fungal infections inside tissues. Neutrophils will phagocyte or ingest microbes to be destroyed and are polynuclear leukocytes, which means that their nucleus is segmented into multiple, up to 5 connected lobules. Their nuclear segmentation is important for the neutrophil cell to produce extracellular traps or nets, which is the action of cell releasing its DNA to trap these pathogens as a last effort to protect the host. Monocytes and macrophages are mononuclear phagocytes. Monocytes are found in the blood, whereas macrophages arise from monocytes and are found in tissues. Specialized macrophages are found in organs like the brain, liver, lung, and spleen, and come from tissue macrophage precursors from the embryonic stage, or hematopoietic stem cells. Both macrophages and monocytes are antigen-presenting cells presenting phagocyted pathogens to T cells capable of activating an adaptive immune response. An important note is that neutrophils, monocytes, and macrophages are all phagocytes. Dendritic cells' main function is to alert and signal to the immune system of foreign invaders. Usually, this cell is considered as a cell that activates the adaptive immune system and presents phagocyted antigens to T cells. Natural killer cells, on the other hand, do what their name suggests, which is to eliminate affected cells, in particular cells affected by a virus. Natural killer cells eliminate cells that supports the MHC1 molecule, a critical molecular component that shows a preview to the cell's peptide production, usually showcasing healthy cells. Cells that suppress their MHC molecule alert natural killer cells that they are viral transcription factors. The virus will attempt to hide itself from natural killer cells by suppressing the MHC1 molecule. Natural killer cells have a similar chemical pathway to eliminate these affected cells, like cytotoxic T cells, that involves the release of perforin and granzymes. Moving on to eosinophils, basophils, and mast cells. All three of these cells share the characteristics of having cytoplasmic granules. These granules contain antimicrobial and inflammatory chemicals. Both eosinophils and basophils are blood granule leukocytes. Eosinophils attack parasites, and their cytoplasmic granules may be harmful to host tissue. Basophils and mast cells can release histamine, triggering allergic reactions and inflammation. Mast cells are derived from the bone marrow, and all of these cells are recruited to inflammatory sites. Cytokines coordinate the immune system response by driving inflammation and activating macrophages and natural killer cells, and as well as other cells. Some cytokines are known as interferons, which create an antiviral state by stopping viral replication. Complement systems, it enhances the immune response. These complement systems actions include opsonization, membrane attack complex, and inflammation. Opsonization occurs when the C3b coding microbes flags the pathogen and is phagocyted and eliminated. Membrane attack complexes are formed by C6 through C9 proteins forming pores into the microbe, killing the cell. And the complement system will recruit immune cells such as cytokines C3a and C5a, which will cause inflammation. Inflammation and antiviral state. Inflammation presents itself physically as redness, heat, swelling, and pain. This is caused by increased blood flow, bringing immune cells to the site of an infection. And the antivirus state is activated by interferon alpha and beta, which is released by infected cells. These interferons will inhibit the replication of viral genetic material by turning off translation, preventing surrounding cells to become infected.
Okay, so how do innate immune reactions stimulate adaptive immune responses? Innate immunity and adaptive immunity work together to provide a line of defense for our body against pathogens and diseases. One of the main functions is to present antigens. This is where the dendritic cells and macrophages are activated by antigen-presenting cells, APC, which come from the innate immunity to activate adaptive immunity. Activated APCs migrate to lymph nodes where they meet T cells, presenting the processed antigens to them. After an APC ingests an antigen, the broken down peptide fragment is then displayed on the cell surface, binding to MHC molecules. These MHC molecules initiate immune response. The T cells recognize the antigens. MHC1 is specific for all cells, while MHC and presents antigens to cytotoxic T cells, while MHC2 is present to helper cells, initiating immune responses. The innate immunity also secretes cytokines, messengers that signal there is a threat, which in turn initiate adaptive immunity response. The complement system also plays a role in supporting adaptive immunity by opsonization of pathogens and lysing infected cells through MAC, membrane attack complex. PAMPs and DAMPs act as signals that alarm the innate immune system by binding to specific immune receptors, PRRs, found on the immune cells. This then initiates an inflammatory response, linking the two systems to the adaptive immune system by activating APCs and promoting the development of specific immune responses against pathogens or damaged tissues within the body.
Thank you. So our sources.
Really. I don't get why these are always about planets.